So next is DNA translation, in which RNA makes proteins. We already discussed that RNA makes protein. Which kind of RNA? It is mRNA makes protein. So in transcription we have seen that how mRNA is formed from the segments of DNA, and here this is our mRNA. This mRNA code for the specific protein or specific amino acid in the protein. So this mRNA have different code for the sequence of amino acid in the polypeptide or proteins. So this nucleotide sequence is responsible for this code, and it direct the amino acid sequence to the formation of polypeptide chain in a polypeptide chain. Now this is the genetic code, which is combination of three nucleotides. First one is A, U and G. This from one code, or we can say is a codon, which is responsible for the formation of one amino acid. Okay, so genetic code is a combination of three nucleotides. Okay, here first nucleotide, second and third nucleotide, which make the codon, which form the amino acid, particular sequence of amino acid. So this AUG stands for it is responsible for the code for the methionine, amino acid methionine. This AUG codon is code for the one amino acid known as methionine. Likewise, another codon, this kind of other codon are responsible for the different amino acid, for the formation of different amino acid. Now one scientist, and name of the scientist is H.G. Corona method, produced the method by which they synthesize RNA molecule on the basis of defined combination of base, which means they produce the mRNA having a combination of base pair which is our own interest. So the scientist, name of the scientist is Corona. Silent feature of the genetic code. So these are the codes present on the mRNA known as genetic code, and the silent features of this genetic code are, these codes are triplet. So this is triplet. This one is a 1, 2, 3. Likewise, it is code for it is a code made up of 3 nucleotides. So it is called as a triplet. And there is 61 codon, which is code for the 20 amino acid. Different type of 20 amino acid present in our body. The 61 codon stands for it is a code for the amino acid, particular amino acid in our body, and 3 codons are not code for the any kind of amino acid, and these codons are known as stop codon. This can be asked in examination that what is stop codon. So stop codon is the code which does not code for any amino acid out of 20. So these codons are known as stop codon. This code is specific for the particular amino acid. For example, this AUG is code for methionine, which means the AUG codon will only code for the methionine, which means if AUG codon is present in a mRNA, it will produce only methionine here. Methionine. Okay? If AUG codon is present on mRNA, it will produce only methionine, not other amino acid. That's why it is known as codon is specific, means it is a specific for each amino acid. For some amino acid are coded by more than one codon, means this is methionine which coded by the AUG codon. But there are certain amino acid which is coded by different codon. For example, UU, A, TT, AA or U. It is possible that both the codons are responsible for the production of particular amino acid. Okay. Now, codons are read in mRNA in a continuous fashion without punctuation. Means these codons are present on the mRNA which read by the ribosome or tRNA in a continuous fashion. Means this codon is first it will read this codon, then this codon, this, then this, then this. Likewise, it is continuously read by in a continuous manner. It is by without any failure. Means it is not like that first they read this codon, then they left this codon and read this codon. It is not happen in life. It is read in a continuous manner. So the codes which is present on mRNA, which is responsible for the formation of sequence of amino acid in a polypeptide chain. So, and next one is code is universal. Means this code are universal. For example, this AUG is a one codon which code for the methionine in a human. So it is a universal, means this AUG will code for the methionine in all the organism, whether it is a prokaryotic or whether it is a eukaryotic or human or another organism. This AUG will responsible for the formation of methionine in every organism. So this universal code property is we can use in a mutation, it is in a genetic engineering, where we can use this code, the code of bacterial code for the particular formation of protein, we can use in another organism to produce another kind of amino acid and another kind of different kind of protein, which is ultimately useful for us. Now, this AUG is known as initiator codon. This can be asked in exam that which codon known as initiator codon. So AUG is known as initiator codon and it is code for methionine. This AUG code known as initiator codon, which initiate the translation, and methionine, it is code for methionine. This is a very important that this nucleotide present in a codon is a specific. If a failure of particular nucleotide to form in a particular sequence of mRNA, it will lead to the formation of another amino acid. As a result of this, very serious kind of diseases can be occur in a organism. So let us see what kind of diseases can be formed in a organism. If the mutation occur in a one nucleotide, here, let's see here, this is gene. This is only one nucleotide which change, okay, with the another nucleotide, like yes, A or T, anything, okay. So what will happen? There is one disease called sickle cell anemia, which is caused by the change in only one nucleotide. It is changed in a 6th position of बीटा ग्लोबुलिन चेन. मीन्स जो बीटा ग्लोबुलिन चेन होती है उस पे 6th पोजीशन पे एक वन न्यूक्लियोटाइड विल चेंज एंड इट रिजल्ट इनटू फॉर्मेशन ऑफ वेलिन in the replace of glutamate. So what happened in a sickle cell anemia? At the 6th position of beta globulin chain, one nucleotide is changed. As a result of this, valine form as a place of glutamate. It results into the change in the structure of RBC in a sickle form, so which carry the less amount of oxygen, and this is known as sickle cell anemia. Okay. So up to this, all right. Now we will discuss about how mRNA is read by the ribosome. During early time, there are many scientists who said and proposed different theories for the how ribosome read mRNA. For example, this is mRNA. Having particular codons. In early time, some scientists said that it is read by this mRNA having particular packages for each amino acid. Said that this is mRNA have package for each amino acid. So like this amino acid will bind here, this here, and peptide bond will form in between them. But it is actually not have sense, that much sense. It does not have sense that it is not present in a life. So we know that life is a very smart, and it does not go this way. It go very simple manner. Later, Francis Crick come with the new hypothesis or new theory that one adapter molecule is there in between mRNA, and we know that the protein is synthesized by ribosome. So ribosome and mRNA, there is one adapter molecule present, and that adapter molecule later found that it is a tRNA. So tRNA is known as adapter molecule. Why this tRNA known as adapter molecule? Because they have anti-codon loop. What is the meaning of anti-codon loop? For example, here A, C, G. It is a code, okay? This is code. So tRNA have anti-codon loop, means they have one side where it is this is tRNA, have two sides. First one is have anti-codon side, which means they can bind with this codon of mRNA having anti-codon complementary base pair. For example, here it is C nucleotide, it will bind with the uracil, then C will bind with the G, and G will bind with the C. So this tRNA have two different sides, one for anti-codon loop and another one for the particular amino acid. So this tRNA code for the particular sequence or for each codon, there is a specific tRNA, and this tRNA carries different kinds of amino acid. Now what will happen? This is amino acid. Now this tRNA first have anti-codon loop, then amino acid acceptor. Here this site known as amino acid acceptor, and we know that we have already discussed that this tRNA is specific for each amino acid, means specific amino acid will bind with the tRNA, and this tRNA will bind with the particular sequence on the mRNA. Okay, this is mRNA, this is tRNA, this is amino acid. Again I repeat that tRNA bind with the specific amino acid. For example, if it is have a code for AUG, then this tRNA bind with AUG, means methionine. If it is code for the AUG codon, then this tRNA have amino acid called methionine. Okay. Likewise, this tRNA have two different sides. First one is a anticodon loop. This is known as anticodon loop, and another side is a amino acid acceptor. Okay, so this is about a tRNA, which is adapter molecule found in a translation. During translation, what it indicate? This tRNA, what indicate? This RNA is a work as an enzyme in translation. During translation, that indicate that in a primitive time when there is no life on earth, most of the cellular activities is carried out by this RNA. And we know about the RNA world, where RNA is the main component in a cell and perform all the function. This is the evidence. This tRNA catalyze for the formation of a particular amino acid or protein synthesis is evidence that in a primitive time on earth, there is all the function is carried out by the RNA only. So this is about tRNA. Now we will discuss about another process of central dogma of molecular biology, that is translation. What is a translation? Translation is the process of making प्रोटीन फ्रॉम द आरएनए नोन एज ट्रांसलेशन. मीन्स आरएनए में से जो प्रोटीन बनता है उस प्रोसेस को हम लोग ट्रांसलेशन बोलते हैं. अगर उसकी डेफिनेशन देखें, इफ वी डिस्कस द डेफिनेशन ऑफ ट्रांसलेशन, दैट मीन्स द पोलीमराइजेशन ऑफ एमिनो एसिड टू फॉर्म पॉलीपेप्टाइड नोन एज ट्रांसलेशन. ट्रांसलेशन मीन्स जो एमिनो एसिड है उसमें पॉलीपेप्टाइड चेन में पेप्टाइड बॉन्ड बना के प्रोटीन बनाने की प्रोसेस को अपन लोग ट्रांसलेशन बोलते हैं. मीन्स ट्रांसलेशन इज द प्रोसेस ऑफ polymerization of amino acid to form protein or polypeptide. So how this process take place? Let us discuss about this process. We know that in transcription one tRNA is formed inside the nucleus. From this nuclear pore, this tRNA will come out from the nucleus into the cytoplasm. Okay, this is tRNA, this is mRNA, which form inside the nucleus, will come out from the nuclear pore into the cytoplasm. Consider this is the nucleus and this all the regions are cytoplasm. Now we are in cytoplasm. Okay, suppose we are in now we are in cytoplasm. What happened in translation? In translation, when tRNA come outside in the cytoplasm, ribosome molecules or ribosome joined with the mRNA. Here this is mRNA and ribosome is bind with this tRNA for the translation. Okay, and this ribosome have particular site for the tRNA molecules. What will happen first? Ribosome